Hey, I'm an ant. You know, let me tell you a little bit about our life. A lot of people don't know about it. We move leaves all throughout the forest. We bring them from trees, from vines, from anything with a leaf, and we take them to our colony. We use them to grow fungus, and then we eat the fungus. It gives us protein. Look around. You can see all my sisters working. There can be up to 8 million of us. It's a pretty great place to be. We're actually responsible for decomposing up to 20% of the leaf matter in the forest. That's pretty important work. Hey. What are these guys doing? Oh, we got humans. Humans near the nest. Well, I think they're trying to figure some stuff out about us. There's some talk on the street, on the forest floor, that we're messy, that we're inefficient. And I think these guys might be coming to do a little bit of work to figure out if the rumors are true. Reports from the soldier ants are in, and it appears that humans were trying to learn more about us. We witnessed them watching us, taking observations, and doing a little reconnaissance. And now, our spies have told us that they're abuzz with questions.
What I love about science is that when you start that process, you end up generating more and more and more questions.
We've been trying to narrow down exactly what we're going to study, and I think we're on the right track. It's going to be cool to design the next phase of this project.
And really what we're finding is that we have more questions than we're capable of answering, but they're all tied together through a sort of unifying theme of the ant-to-leaf relationship.
So, what did they decide to do?
Let's set up 2 types of experiments. In the first, we'll collect the cuttings ants are dumping at their nests. We'll measure the area, mass, and record freshness of those cuttings. Let's also collect leaf fragments that ants are actually carrying. Those will probably be different than the ones they later reject. We can measure the same parameters and also time their rate of transport to try to figure out their efficiency.
The humans head out to the field.
We're coming over to check on the mesh that we put down on all the ant colonies.
And if you look, you see there's this great accumulation of leaves that were clearly dumped right outside their colony. And so from here what we're going to do is we're going to collect up these leaves that are on the surface of our mesh and see if we can find a unifying characteristic of each of these leaves that can help us determine why they were deemed, unacceptable for being brought into the colony itself.
Caught in the act.
These 2 ants, maybe more, are working together. And so this is a leaf that the colony decided wasn't okay. And we don't know why. But we'd like to know why.
20 minutes later.
Alright, so what we're going to do next is gather up our screens and shake them out into the Ziploc bag so that we can get an accurate count of the biomass that's in there.
And see what is the rate at which they discard leaf cuttings at their actual colony. I'm super excited.
Back at the lab.
We have brought our leaf samples back to the lab, and we had a discussion as a group, and we decided that the best way to collect data here is to sort each of these leaves onto a piece of transparency.
So come with us to the leaf analyzer and we'll see what we got. This is a leaf area meter, and it's what we're going to use to see how much total surface area the ants disposed of outside of their mounds. So here we go with our first sample.
This is the last step in our process. These are ovens in this room, and we have one oven in particular that we're going to use. So what I'll do is open it up, and I'll lay these leaves, or the leaf cuttings, on a paper towel that'll go inside the oven, and then I'll turn it up.
Now that they know what we're dumping, they find out what we're carrying.
Here we are assembling our LASDs. That would be leaf ant separation devices. This is very high tech, very fancy. We might even patent these. That's what I heard.
Brace yourself. You need some highly sophisticated equipment. You need a zippy bag and scissors. And what we're doing, this was Galit's fantastic idea, is we're cutting a hole in the very corner of the bag that's big enough for an ant to fit through, but not large enough for a leaf fragment to fit through.
The humans are coming! The humans are coming!
Pinchers ready, hunters ready, go.
Twice! They did it twice! And this time at another colony!
Really important in science that you have replicates, so if we just did it once over there and got some great number, we don't know if that's just true for that colony over there and for nowhere else in the world, but if we can at least add one more colony, then the answer that we get, if it's similar, tells us something about leafcutter ants in general, so that's pretty cool.
After the humans collected our cuttings, they took it another step, and they looked at video of each of us crossing a path, so that they could figure out how many there were moving. Oh, the humanity!
We predicted we would have more green than brown in our collected leaves and more brown than green in our dumped. Well, if you take a look at our actual data, you can see that that pattern really does hold true. There's more brown than green in the discarded leaves at the colony, and we think the reason for that is that the ants are letting those leaves sit inside the colony for a little while longer before they decide to dump them.
Based on the humans' measurements, they concluded we ants move 102 leaves per minute along a single trail, but only discard 0.07 leaves per minute at each dump. That means we ants are around 99% efficient.